Hi, I'm paoladama, founder and CEO of NAMina Bio, a resident company in biolabs at NYU Langone in New York. NAMina Bio is developing human-relevant translational research programs using advanced 3D cell culture, major tumoroids, dynamic culture systems, and analytics-supported study design. The goal is not to replace what research teams already do, but to help them add a more predictive human-relevant layer before advancing to expensive or low predictive downstream studies. This video is meant to introduce how NAMina can support pilot study, collaborative programs, and early translational decision-making. Here I explain the broader reason why NAMina is focused on human-relevant model systems. Drug development still faces major translational gaps, especially when transitional models fail to predict human biology. New approach methodologies, including organoids, 3D models, microphysiological systems, and advanced in vitro platforms are becoming increasingly important because they can provide more human-relevant evidence earlier in the research process. For companies and translational researchers, this creates an opportunity to de-risk early decisions before investing in larger or more expensive studies. NAMina is not only offering predefined research programs. Our model is broader. We help researchers and companies access, adopt, and integrate human-relevant technologies into their own workflows. This can include a collaborative pilot study, but it can also mean helping a lab select and implement the right technology. For example, dynamic culture systems, bioreactors, ECM matrices, 3D culture systems, organ-on-a-chip tools, synthesis capabilities, or analytics-supported readouts. The value of NAMina is the integration layer. We help connect the scientific question with the most appropriate technology, experimental design, and translational interpretation. This is the practical entry point. We usually recommend starting with a focused pilot study rather than a large complex program. A pilot can be designed around a specific biological question, such as whether a compound behaves differently in a mature 3D system compared with a standard model, whether a dynamic culture condition changes response, or whether a combination strategy has a stronger activity in a more human-relevant context. The pilot framework keeps the study small, interpretable, and useful. It allows both sides to assess scientific fit before expanding into a larger collaboration. This infrastructure is designed to support different levels of engagement. Some groups may want NAMina to help design and execute a pilot program. Others may want to bring specific technologies into their own laboratory and use them with the NAMina's assistance, of course. Our portfolio includes both services and products through a curated network of technologies: bioreactors, dynamic culture platforms, ECM systems, 3D model tools, microphysiological systems, synthesis capabilities, and analytics. This makes NAMina useful not only as a service provider, but also as a practical partner for technology adoption and translational workflow design. In addition to static 3D culture, NAMina is also building programs around dynamic culture systems. These systems can introduce flow, perfusion pressure, and more physiological relevant exchange conditions. This is important because many tissues are not static in the human body. Tumors, barrier, vascularized tissue, and organ-on-a-chip systems may behave differently when exposed to dynamic conditions. So for collaborators, this opens the possibility of testing whether biology, drug response, toxicity, or barrier function changes under more realistic experimental conditions. The analytics layer is designed to help structure and interpret complex biological data. As models become more advanced, the amount of the complexity of data increases as well. NAMina's goal is to use analytics to support better experimental interpretations, biomarker discovery, response profiling, and cross-model comparison. This does not replace biological expertise. Instead, it helps organize the evidence so that researchers can better understand what the model is telling them and how it may inform the next experimental or translational step. The final slides here summarize the type of collaboration we are looking for. NAMina is interested in working with researchers, startups, platform companies, and translational teams that want to test a focused biological question using human-relevant models. A good first step could be a short discussion to identify the scientific question, the most appropriate model system, the readouts, and the expected decision point. From there, we can define a small pilot that is scientifically meaningful, realistic, and aligned with the collaborator's goal. If you are interested in NAMina, please contact me and visit my website. Thank you for your attention.